This is a video lab report for Lab 5. I'm Ivan Lebedev.
And I'm Dan Brandon. The primary objectives of this lab were to observe the circulatory system with particular emphasis on biological buffers, and to observe the respiratory system with emphasis on vital capacity.
The circular system's primary function is to transport nutrients, gases, waste, and hormones to various body tissues. The body is sensitive to changes in acidity, so it has developed an advanced and reliable buffer system known as the bicarbonate buffer system, which keeps the pH between 7.0 and 7.8 in mammals. Outside this range, it will do irreversible damage to the tissues.
The second important system involved in this experiment is the respiratory system. This system is vital for gas exchange, where oxygen is introduced into the blood and CO2 is removed. And lungs have a large surface area and in turn a large volume. The vital capacity is the maximum amount of air a person can expel after a maximum inspiration or inhale. The value of vital capacity is a good measure of a person's lung capacity. To keep the lungs from collapsing, considering drastic pressure changes in the lungs, surfactant is released from the cells, lowering surface tension, which helps in preventing collapsing of the lungs.
In this lab, 3 primary experiments were performed. The calcium hydroxide experiment, where room air and exhaled air was bubbled through a solution of calcium hydroxide. When carbon dioxide reacts with calcium hydroxide, the solution turns cloudy, so it is predicted that the exhaled air will turn the solution cloudy more quickly.
The second main experiment we performed in lab was phenolphthalein experiment, where room air, exhaled air at rest, and exhaled air after exercising was bubbled into the solution. When CO2 was bubbled through, carbonic acid formed in the solution, which increased the acidity, turning the solution clear, as can be seen in the left beaker in figure number 4. To find out how much CO2 was put through the solution, ammonia was added dropwise to lower the acidity until the solution turned pink again, as you can see from the right beaker in figure 4. It is hypothesized that there will be more CO2 in the exercising air than the resting air. The exhaled air will require more drops of ammonia than the room air to turn it back to pink.
The final important experiment was the vital capacity experiment, in which each participant inhaled as much as they could, then exhaled as much as they could into a spirometer. The values were recorded, then compared to what each male or female should be capable as a function of height and weight. It is predicted that individual vital capacities will be within the normal range. All the materials were provided by the Department of Biology, and methods were presented in the laboratory manual provided on MyWPI. There were no deviations from the manual. Results for the calcium hydroxide experiment showed that exhaled air turned the solution cloudy, whereas the room air had no change in the solution's clarity. For the vital capacity experiment, it was found that the class average was 3.4 liters, with a standard deviation of 0.79 liters. Both Dan and I had vital capacities within 2 standard deviations, which fit underneath the normal curve. The figure to the right is a histogram, where bins are vital capacity ranges, and frequency is the number of students within each range. For example, only 1 student had a vital capacity between 2 and 3 liters, while 32 students had vital capacities between 3 and 4 liters, and so on. Considering the class has more females in it than males, and females tend to have smaller lungs, it is expected that the average will be closer to the female average than male average. Other deviations from normality can occur due to particularly tall, short, heavy, or white individual, as vital capacity is dependent on these factors. In the individual case here, as predicted, the vital capacities were within the normal range, as neither Dan nor I are excessively tall, short, over, or underweight.
For the phenolphthalein experiment, as you can see from Table 1, it was found that room air needed an average of 5.8 drops of ammonia to normalize the acidity of the solution. Exhaled air from rest needed 79.4 drops, and exercise air needed 123.4 drops. These values were normalized to room air and represent the class data. The individual results showed 23 drops for room air, 93 drops for exhaled air from rest, and 233 drops for exercised air. These values differ from the class data, and there's much variation among individual groups. This can most likely be attributed to the difference in individuals. Some may have exercised harder than others. Some may just naturally expel more carbon dioxide than others. Following an analysis of variance test, what can be certain from our data is that the amount of carbon dioxide expelled is significantly different for each set of trials. That is, the amount of CO2 expelled after exercising is significantly higher than exhaled from rest, which is significantly higher than room air. An analysis of variance was performed, and it provided us with a p-value of 0.04. Because this value was below 0.05, we were able to reject our null hypothesis that there was no difference between the groups. And it can clearly be seen here that it is statistically significant that each group is different. And judging by our data here in Table 2, we can say that our hypothesis holds true.